Dear everybody, this is the late talk of today, but you know, we always put the best people at the end, otherwise we are running the risk that people are disappearing. Tom Seeley, he's one of the good friends of Danish Beekeeping. He was already there when we had 150 years anniversary, and it was in the small city or town of Ringe, where Tom actually made 3 very, very nice talks at that time. I have been so lucky to meet you, Tom, on different occasions, like you said on the Irish Beekeeper conference last year, where we made a talk right after each other. And of course, you're one of the hot topics out there right now on what could we call it, natural beekeeping, and you have been working with this for so many years. And I always make fun with you. Do you know that? Tom Seeley, do you know what he did? He was sitting. I see this picture. He's sitting on a chair with a cap on and a small table outside there, and that's the science he has done. And he made all the conclusions on just looking up on the bees. So this is really what I call natural science. Of course, I know this is far more advanced, everything. Tom has made great books out there. I mean, this is the latest one that I got from him, the Lives of the Bees. And then there is another, I call it the secret book. And that's really the best one for me. It's really the honey hunting book that you made maybe several years ago. And I would really recommend everybody go on the internet, Google Tom Seeley and his books, and buy them. Tom, you owe me a beer now next time we meet.
Yeah, that's right.
Yeah. So,
thank you, Flemming.
So, Tom, the stage is yours. We're really looking forward to your talk and are very, very excited. There are approximately 500 people out there watching you, and they are mainly from Denmark, but we have lots of people from Norway and from Sweden, and some from England and some from Germany as well. So we could really call this an international conference. Please.
Okay. Thank you, Flemming. It's always fun to see you, and I have very fond memories of our times together. You tell such great jokes, by the way. Now, quick question for me to ask you at the beginning: how do I advance a slide? Do I just say to you next slide, or because I don't control this? I think, is that correct?
Oh yeah, so we should maybe just you just say next slide, or maybe
just say next slide, and then Sophus will advance it.
Okay, brilliant, great. Well, yeah. So what I'm going to talk about is we'll build upon what the previous speaker, Marla Spivak, was talking about, where she reviewed how bees have been selected to be hygienic and things like that. To get better resistance to Varroa and thus better colony health. What I'm going to do is take a related topic and look at bees that have not been managed by beekeepers, have been living on their own in a forest near where I live, and we'll look at how they're dealing with honeybees. And we'll put this in a broad context of really a different approach to beekeeping. It's what I call Darwinian beekeeping, which is an evolutionary approach to beekeeping. So next slide, please.
Oh, we are working on it.
Okay. There we go.
Okay.
We could go back one slide, please. There we go. Thank you. These are the 2 gentlemen that are figuring heavily in this talk. Lorenzo Langstroth on the left, Charles Darwin on the right. And they were basically contemporaries. You can see they were born in almost the same year. And the reason I juxtapose them is because they are very both important to understanding beekeeping, especially what I call this Darwinian beekeeping that I'm going to be talking about. Next slide, please. One of the things that they had in common was that they both studied comb building by honeybees. Langstroth in the context of inventing the movable frame hive, and Darwin was also fascinated by comb building by honeybees, not to study not to use to manipulate their comb building, but he admired the precision with which they build the regular pattern of cells in their comb, and he called it the most wonderful of all known instincts. Next slide, please. The main reason I juxtapose them today is that they both have insights that can help us with our beekeeping. Next slide, please. For Langstroth, it was, of course, the concept of the movable frame hive. Probably everybody here has their bees living on movable frame hives, as do I. For Darwin, the key thing that he produced that can help us with our beekeeping today is this concept of evolution by natural selection. Let's review what this concept is. Next slide, please. I'm going to illustrate an example of evolution by natural selection by referring to the evolution of pesticide resistance in insects. And as we can see on the left there, is a diagram where we can see at the top a population of individuals before selection, and the color coding there reflects the differences among the individuals in their resistance to a certain pesticide. Some individuals have low resistance, some have high resistance before selection. After selection, after the pesticide's been applied to the field or the population, only the individuals that have high resistance are still around, are still alive. And so the population then, when it rebuilds itself, it's going to consist of individuals that have high resistance. So that's a very simple but clear, I think, hopefully, example of evolution of natural selection, how it produces traits that enable animals, including these insects here, to survive and reproduce. Next slide, please. So, how do we apply Darwin's concept of natural selection to beekeeping? How does it apply to beekeeping? Next slide. We could go to the next slide, please. This concept tells us that everything that honeybee colonies do when they're living in nature is done to favor their survival and their reproduction. And when I say everything, I really mean everything. The amount of brood they rear, the amount of pollen they collect, the amount of honey they store, and all of their other traits that of colonies, they are present in honeybee colonies because they have been favored by natural selection. The colonies that weren't good, for example, at collecting pollen, they didn't survive and reproduce. So it's very important, I think, when we look at our honeybee colonies to understand that they are very well adapted to their natural environment, the environment in which they lived for thousands of years before there were beekeepers. Next slide, please. And here's the main idea of this talk. This Darwinian beekeeping is a set of beekeeping practices that enable honeybee colonies to use their own beekeeping skills fully. So the philosophy or the approach of Darwinian beekeeping is to keep bees in ways that let the bees use their own skills as fully as possible. And the way we can do this is by enabling colonies to live as naturally as possible. Next slide, please. And you've probably heard, you may have heard about Darwinian beekeeping, or you may have heard about natural beekeeping or apicentric beekeeping. They're all pretty much the same idea. The one difference, I think, is that Darwinian beekeeping is science or evidence-based. What I'm going to share with you, the ideas that I'm going to suggest for Darwinian beekeeping are based on what we've learned by scientific investigations of how bees are living in nature. So not guesswork, but actually going out and looking at how bees live in the wild. Next slide, please. And here I want to make a very important point. Darwinian beekeeping is not for everyone. It's not for everyone. Next slide, please. Well, who is it for? Well, first of all, who is it not for? It's not for large-scale beekeepers, which is most beekeepers in the United there are many large beekeepers in the United States. It's not relevant for them. It's maybe not for urban beekeepers, because a key part of Darwinian beekeeping is letting colonies swarm, and that can be a problem. That can create problems in cities. What it is, though, Darwinian beekeeping is an option for small-scale beekeepers who want to produce miticide-free honey and who are satisfied with modest honey crops, only about 25 pounds per hive, or that's 10 kilograms per hive. And I see that, unfortunately, I had the file that I sent you was one I sent before I had converted everything to metric, so I will do that as we go through the talk. So with Darwinian beekeeping, you can harvest about, at least I have, about 10 kilograms of honey per hive. Not a large amount, but some. Next slide, please. Here's another way to contrast or to think about Darwinian beekeeping relative to commercial beekeeping, where the beekeeper would have lots of colonies. It's like the difference between having one backyard apple tree versus a commercial orchard. Maybe an even better comparison is the difference between bird watching and poultry farming. With bird watching, what do? You put out a bird feeder, you put out bird houses, the animals move in, you enjoy them around your home or wherever. And that's very modest. Poultry farming, at least in the United States, it involves large buildings and thousands of poultry. And so it's another way to think of it is Darwinian beekeeping is about pleasure, taking pleasure with beekeeping versus getting a profit. Next slide, please. And one thing I want to stress here is that both of these approaches are valid. They just have different aims. They're for different purposes. Both approaches are valid. I'm not saying Darwinian beekeeping is better. I'm just saying it's appropriate and relevant for small-scale beekeepers who don't want to treat their bees for varroa. Next slide, please. So I'm going to be talking about letting colonies live naturally. And you might be wondering, well, don't honeybees need our help to deal with varroa? Can we just let the colonies live on their own somehow? And the answer, and so let's look at the next slide. The answer to these questions, in my experience, is yes. And I say that because that we can allow bees to live naturally if we do it right. And the reason I say that is because I find where I live that colonies that are living wild in trees and in buildings, they have shown me that honeybees don't always need our help to deal with varroa. In fact, as we will see, the colonies that are living out in nature where I live, they're doing just fine controlling varroa and being able to live. Next slide, please. So what I'm going to show you now is what I've learned, that the wild colonies living around Ithaca, New York have not been killed off. And I'm going to go through this in some detail because I want to make it clear that what I'm proposing isn't something I dreamed up. It is something that the bees have taught me. And the first step in learning from the bees about Darwinian beekeeping was just to see whether the bees are capable of taking care of themselves. Next slide, please. Let me tell you a little bit about where I do my studies. I work at a university called Cornell University. It's based in a small city in New York State, the city of Ithaca. Probably everybody knows about New York City, and when I say New York, people think of New York City, but Ithaca, New York is about a 5-hour drive from New York City on a high-speed highway. It's up in a rural part of New York State. And the reason I show this map is that it illustrates how most of New York State is covered with forests. Wherever you see green in this map, that means that's mostly forest cover of the land. It's because most of New York State is mountainous, and Ithaca is at the base of the part of the Appalachian Mountain region. Next slide, please. Give you a sense of what this part of the world looks like. It's heavily forested. You can see just a shot of the road near my house, and that's my house, and it looks like that, what I'm showing here right now. We've got about 4 feet of snow on the ground. And so it gets very cold winters and very hot summers. Next slide, please. And here's a Google Earth photo that'll give you a better feeling, I hope, for the vegetation and the terrain in Ithaca and those areas around. So you can see Ithaca there, and then there's a box to the southwest of Ithaca called the Arnott Forest, and that's the area. It's a forest owned by Cornell University, and it's the area where I've been following the wild colonies, how they're living for about 40 years. Let's take a look at the Arnott Forest. Next slide, please. As I mentioned, this is a forest that's owned by Cornell University. It's a teaching and research forest, so it's protected. But the other thing I want to stress at this point is this Arnott Forest is just a sample of the forests in this part of the world. So what I'm going to be showing you about honeybees living in this forest applies to other forests all around over vast areas. So, and there are more wild colonies living in New York State than there are managed colonies, many more, based on the vast areas of the forest and the abundance of the wild colonies, which I'll be explaining now. Next slide, please. Here's an image, gives you a sense of what this part of the world looks like. It's forested hilltops and slopes. Down in the valleys, the early settlers, this area was settled in the early 1800s. Farmers came in, many of them Irish immigrants and English immigrants, and they built farms down in the valleys, but the soil was very poor, and all those farms have been abandoned. So now this part of the world is just going back to forest lands. Next slide, please. And one feature of the Arnott Forest is it is the one place in North America where we have data on the wild colony abundance before the arrival of Varroa. Now, Varroa reached us in where I live around 1994. That was I first saw Varroa in my hives in 1994. They probably came up a little earlier than that, but we'll say 1994. And back in 1978, I had gone to the Arnott Forest and measured the abundance of the wild colonies. And what I found in 1978 was 2.5 colonies per square mile, which is basically 1.0 colonies per square kilometer. So keep that number in mind. One wild colony per square kilometer living in these forests. Next slide, please. And so, I knew that there was one colony per square mile back in the 1970s. Varroa arrived in 1994, and my experience was that I lost about 80% of my colonies each winter for 2 winters before I started treating learned that I had to treat my colonies of bees. I was a slow learner about that. So, but it was, in some ways it was good because it showed me just how powerful Varroa is, how good Varroa was, combined with the viruses at killing colonies. So by the late 1990s and the early 2000s, I was assuming, I believed that there were no wild colonies going left out in the forests because I had seen such heavy mortality of the untreated colonies that I keep in my bee yards. And I keep about 60 colonies of bees. So I was losing a lot of colonies of bees until I learned to treat them. But then in 2002, I realized I really should go back to the Arnott Forest and see if it really was true that all of the wild colonies had been killed off. So in 2002, I went back and looked again to find the wild colonies in the Arnott Forest and determine their abundance. What did I find? Next slide, please. Oh, first of all, let me explain how I find these wild colonies, which you may be wondering about. How on earth do you find, how do you find wild colonies living in trees? I do it by a process called bee hunting or beelining, and Fleming has highlighted that little book on that I've written on how to do that. And the process is pretty simple. You go to a field where there are some flowers, and I use a little box in which I capture a bunch of the bees off of the flowers, and I introduce them to a square of comb that in which I've put sugar syrup. And of course the bees like that unless there's a honey flow on, and they fly home, and then they bring their nest mates, and pretty soon you'll have a lot of flight traffic away from your feeding station back to the bee's home. And basically you then work your way step by step down that bee line, and that leads you back to the bee's home. Next slide, please. So the first step is, once you've got bees flying off from your little feeding station, you see what direction they fly home. That's determining the bee line. And then once you've got a clear sense of the bee line, you then move your feeding station step by step down the bee line, getting closer and closer to where the bees are living. Next slide, please. This map on the left illustrates the bee hunting process. This was a bee hunt I did on the campus of Cornell University, where I started my bee line at a flower garden, caught bees on the flowers, and then you can see I did that. Started my bee hunt at noon, and then in a series of steps, each one about maybe 100, maybe 60 meters, something like that, I moved from where I started, and then I moved north step by step, and finally this led me to the bees living in the bee tree. The bees were living, a wild colony of bees was living up in this oak tree. And you might wonder, how do you actually see the bees, given that the entrances are usually very high off the ground? It's not as hard as you might imagine, because what you see up there is the flight. Our eyes are very good at detecting moving objects, and you can see the little bees flying around against the bright sky. Next slide, please. So here's a map of the bee hunting I did in 2002, when I went back to the Arnott Forest to see if I could find any bee trees. And I wish I had a pointer at this point, because what I'd like to do is, do you see this pointer at all? Maybe. Well, basically what I did is I started in the northeast corner of the forest, and I caught bees on flowers there, and some of the bees flew south, which led me to what I've labeled as bee tree A. It took me 2 days to find bee tree A. Thank you, Fleming. Once I'd found bee tree A, I then went back and followed another bee line to the north that led me to bee tree B. And then I shut down my operation at that location. I moved down the road to the west to site 2, caught more bees on flowers there. Some of them flew back to bee tree A, and others bees flew up to the northwest to another bee tree C. And so that's the process that I did. Step by step, I went to different locations in the forest and just tracked down every bee line I got, except for a few bee lines that pointed out of the forest. I didn't track those. And so I surveyed half of the forest, the western half of the forest. I worked my way around the western half of the forest systematically, and I found 8 bee trees, wild colonies living in trees in the forest. And that was surveying half of the forest. Next slide, please. Here are examples of some of the bee tree homes that I found. One was in a white ash tree, and the bees were living in what had been a hole created by a woodpecker going for carpenter ants in the tree. Another one was up in this hemlock tree, pretty high up in the tree. Again, it was probably carpenter ants that originally made the cavity in the tree. So those are just examples of 2 of the bee trees in this Arnott Forest, and it gives you a little bit of a sense of what this forest looks like. It's a little hard to judge the size of that ash tree, but this is a forest that has not really been harvested, not logged since about, I guess, early in the 20th century. So that a lot of these trees are 1 meter or more in diameter at chest height. Big trees. Next slide, please. Here, these 2 images here show us really what's a typical bee tree home of one of these wild colonies. The entrance is high, usually more than 5 meters up. The entrance is small, less than about, that would be less than 20 square centimeters. The nesting cavity is usually quite modest, only about 40 liters. And for reference, that's approximately the volume of 1 Langstroth hive body. So they're living high off the ground with small entrances and in rather modest small nest cavities. So the colonies themselves are small, and we'll come back to that, but that means the colonies are very prone to swarm, and that's of course a natural process for bees. Next slide, please. So to come back to this point, the Arnott Forest was the one place in North America where we had data on the abundance of wild colonies before. That was 2.5 colonies per square mile, and I got the exact same number when I came back in 2002, which was after the arrival of Varroa, 2.5 colonies per square mile, which surprised me and delighted me. But my delight was tempered by the realization that I didn't know if these wild colonies had Varroa. After all, these colonies living in the Arnott Forest are very far from any beekeeper's hives. The nearest beekeeper's hives are about 10 kilometers away. So I thought, well, maybe these colonies are still here because they don't have Varroa. So I needed to find out if the colonies living in the Arnott Forest did or did not have Varroa. How did I do that? Next slide, please. Next slide. To address the question, were the Arnott Forest colonies infested with Varroa, I put up bait hives in the Arnott Forest. And for me, a bait hive, this is the bait hive that I used. It's not what I use now, but what I did is I put up Langstroth hives up in trees, as you can see there. I reduced the entrance size, and these were very attractive to the wild swarms. And of course, once bees moved into a Langstroth hive, I could check the colony very carefully for mites. And what did I find? Next slide, please. Well, in the summers of 2003 and 2004, I caught 11 swarms in the Arnott Forest. And I let the colonies once they moved into a bait hive, I would leave the bait hive up in the tree. And I could climb up with a ladder and open up the hive and inspect the colony. And what I found is that every colony had Varroa. It came with Varroa. And over the years, as I say, I left the colonies just living in those bait hives. One of the 11 colonies came down with chalk brood, so there was a little chalk brood also in the population. None of the colonies showed evidence of American foul brood or European foul brood. So the key thing here was that these aren't not forest colonies are actually infested with Varroa, and yet they are surviving. And what I've shared with you here is what I found in 2002. We resurveyed the colonies in 2012 and found the same pattern of results. And I've caught swarms who are not forest colony over the last summer, and they still have the Varroa. But the colonies are still surviving. So that's really sets up the stage for showing, well, it shows us that these bees know how to deal with Varroa. Next slide, please. So the obvious question now is, how are the wild colonies controlling Varroa? They have two things going for them. They have good genes and they have good lifestyle. And so we're going to go through both of these things. And with respect to good genes, as I'll be showing you shortly, these bees are very good at killing and biting the adult Varroa, and they're also very good at uncapping and recapping brood cells that are infested with Varroa. And with respect to lifestyle, they have several things that help them deal with Varroa. One is they swarm every summer, and that creates a brood break. And another thing that they have going for them is that their nest cavities are small. That means their brood nests are small, so there's relatively little brood for Varroa reproduction. And that's especially true in late summer because these colonies, their brood nests get small rather early in the summer. By August, they're filling up their nest cavities with honey, and the brood production goes down. So that also reduces the availability of brood cells for Varroa in late summer. We'll look at these things in more detail now. Next slide, please. And now we go back to Darwinian beekeeping. And this is the framework for thinking about Darwinian beekeeping. It's the difference between the original environment of honeybees, and this is the environment in which wild colonies have lived and still live. Now they live in nature, versus the current environment of honeybees. And this is the environment in which managed colonies live. And I suspect, I don't know a lot about Denmark, but I suspect that the current environment is the environment in which most colonies are living in Denmark. But I would be very interested in learning whether there are some bees living wild in Denmark as well. Next slide, please. So our goal with Darwinian beekeeping is to enable managed colonies, insofar as possible, to live again in their original or natural environment, but not when they're living in trees, but when they're living in beekeepers' hives. Because we as beekeepers, we can manage colonies however we want. We can give them a very natural life, or we can give them a very unnatural life. And Darwinian beekeeping is just saying, hey, one possibility is to, if you want to do it this way, here's what you do to let bees live as they do in nature and allow them to use all their own skills at dealing with Varroa. Next slide, please. So what I'm going to do now is I want to compare point by point the environments in which wild colonies live versus managed colonies. So we're going to just go through a number of slides here where we compare point by point the original environment, the natural environment of honeybee colonies, and their current environment when they're kept by a beekeeper, like myself. In the original environment, colonies are genetically adapted to their location. And at least in North America, most beekeepers' colonies are not genetically adapted to their location. And why is this? Next slide, please. Well, in North America, beekeepers buy their bees from all over creation. In the beekeeping journals, magazines here, there are advertisements for bees from Hawaii, or from Georgia or California or Texas. And those bees that are produced in Hawaii probably are very good bees for Hawaii, but they're not very good bees for New York State or Marlis vivax, Minnesota. But they're so it's important if one wants to do this Darwinian or natural beekeeping to work with colonies that are well adapted to your location. I'm not sure, I'd be interested to know how it works in Denmark. Whether people are buying queens, beekeepers in Denmark are ordering queens from Italy, for example. If so, then I suspect those colonies are not as good as your locally adapted bees. Next slide, please. Second comparison between original environment or natural environment and current environment is in the wild, colonies live widely spaced. In under-managed situations, colonies live crowded in apiaries. That's pretty obvious. Next slide, please. The third point of comparison is that in the wild, colonies live in small nest cavities, as we've seen, about one deep hive body. And that means they swarm freely. And the distribution you see at the bottom in this slide is the nest cavity volumes of 21 wild colony nests where I found the bee tree, collected the nest, split open the and measured the volume of the cavity that the bees were living in. And I did that by removing the bees and the combs and then refilling the cavity with sand. So I could get quite an accurate measurement of the volume. And you can see most of them were about the size of one deep 10-frame hive body. Some were a little smaller than that, some were a little larger, but around one deep 10-frame hive body. And what's on the right illustrates, at least in North America, what is a typical beekeeper's hive, where colonies live in these supersized nest cavities. And I show this because this is an illustration from one of the bee supply catalogs, which says this is the best and most complete wooden kit. It looks like a real, it's very different from how bees live in nature. Let's put it that way. Next slide, please. A fourth comparison between the original environment and current environment is in nature. The nest cavity walls are thick, often 20 centimeters or more thick, and they're coated with propolis. And this image that you see on the left is a photograph of the wall of a nest cavity, and in the lower left portion of the nest cavity, you see an area of the nest cavity wall where I chipped off the propolis, and you can see pretty easily why the bees are covering these cavity walls so thoroughly with propolis, because the wood is porous and fibrous. It is it's produced, that rough surface is produced by the decay that produced the tree cavity, and it's a wonderful site when damp for bacteria and other microbes to thrive. So the bees seal off the nest cavities with propolis. And in contrast, in the walls of our hives, they're usually made with planed lumber, and the bees don't put much propolis on them. In this case, the image shown here on the right, the bees have put propolis on the walls of this hive because the propolis collection screen material had been stapled over those walls. But between you can see that without that propolis collecting screen, the bees would have put very little propolis on the walls, and I'm sure that's what you experience too. Next slide, please. Another difference is the amount of drone comb. In the wild, colonies build drone comb freely, and they produce lots of drones. And when I say freely, in these wild colony nests that I dissected, I found that the bees were devoting 15 to 20% of their comb area to drone cells, to drone comb. Most beekeepers I know, and what I was taught, was to discourage drone comb construction, because you don't want your colonies, I was taught, you don't want them to produce lots of drones. Drones don't add to the productivity, the honey production of a colony. So most beekeepers don't allow their colonies to build much drone comb. In nature, they do. The bees pass on a colony of honeybees passes on its genes by producing drones and by producing swarms. And so colonies that produce drone comb produce drones and are more genetically successful than those that do not. Next slide, please. Another comparison between nature and current environment is the nest entrance is high off the ground, on average 8 meters up in the Arnott Forest. This is me down at the bottom of the slide. That's me. I'm about 2 meters tall, not quite. And there's a typical nest. That red arrow points to the nest entrance, high up in the tree. As I say, on average, in the wild, the nest entrances are very high. Whereas in a beekeeper's situation, the nest entrance is low. It's convenient. Our hives are at ground level, so we can work them. You might wonder, why do bees build their nest entrances so high? Well, we'll come back to that, but in the Arnott Forest, a lot of it has to do with being cryptic with respect to black bears. There are black bears in the forest, and if a bee tree blows over, the black bears do their best to open up that nest and kill and get the honey. So the bees pretty well do a good job of hiding their nests from the bees by nesting high off the ground. Next slide, please. Another difference between the natural or original environment is the colonies have diverse pollen sources. Many times in a current environment, that's still the case, but there are some situations, like in California, these almond groves, the colonies have very low diversity or non-diverse pollen sources. So that's a big difference. And as with most food things, diversity of foods is better than uniformity of foods. Next slide, please. And a big difference in the wild, colonies are not treated for diseases. In current environment, beekeepers, most colonies are treated for diseases. And as we will see, when the colonies are not treated for diseases, the bees can evolve resistance to Varroa. It requires a whole population of colonies to not be treated for diseases. And we'll come back to that. But of course, when the colonies are treated for diseases, they don't have to, a lot of colonies without the right traits can persist. Next slide, please. So that's summarizes the comparison, or concludes the comparison I wanted to draw between how colonies live in nature versus how they're kept in beekeepers' situations. And again, the goal with Darwinian beekeeping is to put managed colonies back into the honeybee's original environment. This is the environment to which they're evolutionarily adapted. Or in other words, our goal is to enable managed colonies to live more naturally. And the colonies will make less honey, but they will have better health. So you can think of it with honeybees, there's a kind of trade-off between honey and health. And we beekeepers, and I'm a beekeeper, we can try to get both if we can, but it requires a lot of medications, treatments for the mites these days. It used to not be so hard. Used to be able to do both, but now it's harder. Next slide, please. What I want to do now is go through, I think it's about 8 guidelines for Darwinian beekeeping. These are techniques that I've worked out over the years that work for me where I live. I don't know if they'll work for where you live, and that's going to depend on a lot of things, which we'll be going through now. But one thing that is always going to be true and is important wherever you are is to keep bees that are adapted to your location. And the way you can accomplish this is you can rear queens from your best survivor colonies. And by a survivor colony, I mean a colony that has shown itself as being able to survive without being treated for Varroa. Another way to get bees that are adapted to your location is to capture swarms in bait hives set out in remote places away from any beekeepers' hives. And I don't know if you have such places in Denmark, but I know Denmark is a is a very complicated geography, and so maybe there are some remote locations out on some islands. And I think you have one island where, in fact, the bees are, it is a treatment-free island, I believe. I'd like to learn more about that. And another possibility is to purchase queens from a queen breeder who produces locally adapted queens and ideally does not treat for varroa. Again, I hope that's available in Denmark, and maybe you can tell me. Let me give you an example. Next slide, please, of where I compared the colonies that I established with non-local versus local queens. And I did this by my non-local queens were queens that I bought from a commercial queen producer in California called Oliveras. And my local queens were queens that I caught in my bait hives, which were, of course, in New York State. And what I did is I set up two groups of colonies in the same location. Two groups of colonies were placed along the same tree line, as you can see there. And each colony was started out as a two-frame nuke. I took 2 frames of bees and brood from other colonies, and I put in each hive 2 frames of bees and brood, and I gave them a queen of one of the 2 types, either the Oliveras queen or my bait hive queen. And so the colonies were set up in June 2019, and I just left the colonies all alone all summer. It was just as if the swarm had moved into those hives. And then in October, I went back and I measured each colony's mite level using the sugar shake test. And that's the end of our summer when the colony has very little brood. And then I, again, just left the colonies there and all winter, and I assessed the colony's winter survival in April 2020, the following April. Next slide shows the results, I think. First, we're going to look at the results of comparing the mite levels in the non-local, the California, and the local, the wild colonies caught in New York. And you can see the mites per 300 bees for the California queens were 17, 10, 4, 24, 9, 5, and 18, whereas the mite counts in the colonies with the bait hive queens, the wild colony queens, were 0, 3, 22, 4, 5, 1, 1, 1. So that looks quite different. And what about the colony survivorship then over the winter from the winter of 2019 to 2020? That's in the next slide. Wherever you see a yellow with a line through it, that means the colony didn't survive that winter. So of the Oliveras, the colonies headed by Oliveras queens, they were big, strong colonies going into winter, and they had a good supply of honey. But only one of them survived. The only one that survived was the one colony that had a low mite count in October. With respect to the bait hives, it's just the other way around. They all survived except one, and that was the one that had a high mite count in October. So the difference in survival was the difference between 14% and 85%. And this is a good, I think, a good example of the power of getting bees, if you can, that are both locally adapted to your climate, your location, and also are naturally selected to have resistance to varroa. Because the bait hive queens, the colonies headed by queens from the bait hives, except for one of them, they were very good at controlling the varroa. Next slide, please. So the first guideline was to get locally adapted stock that's got good resistance to varroa. Second guideline is to house your colonies in small hives. Remember, this is again based on knowing how colonies live in the wild. And for me, when I do Darwinian beekeeping, I get one deep 10-frame hive body, and if I want to get some honey from the colony, I'll put one shallow honey super over a queen excluder. And I will take that honey super off as early as I can in the summer, so that the colonies are really, most of the summer, are just living in one 10-frame hive body. And to illustrate the importance of this small hive, I did an experiment, or to find out how important it was. I did this experiment where I set up 2 groups of colonies. There were 12 colonies, which I called simulated wild colonies, because each colony was just put in a 10-frame hive body, and the colony was started like a swarm moved in. And nearby, only about 100 meters away, I set up another 12 colonies, and each of these colonies was put in a large hive, 4 deep hive bodies. So they made lots and lots of honey. The colonies got really big and really strong and made lots of honey. But what happened about the Varroa counts in these colonies? Well, we'll see. First of all, look at the survival. 2 years later, the colonies were set up in the spring of 1 year, and I just let the experiment run for 2 years. After 2 years later, 10 of the 12 colonies living in the small hives, simulated hives, they were still alive. So 83% survival after 2 years. Only 17% survival in the colonies that were put in the large honey production hives. And why is that difference in survival? That's shown in the next slide. It has to do with Varroa. These are the mite counts over the 2 years. The colonies were set up in July 2012, and for the first year, because the colonies started out with quite low small colonies, their mite counts didn't get out. They didn't get in. The colonies did pretty well the first summer, but so the second summer, May 2015, you can see there were still, the mite counts were pretty low for both groups of colonies. But then the colonies in the large hive treatment, in that second summer, their mite counts just went up and up and up, but they stayed low in the small hive treatment. And that's a lot has to do with the swarming by the small hive colonies. And that's what happened just by changing the different sizes of the hives. The colonies, the queens came from the same source. I should have explained that. All the colonies were headed by commercially available queens. So just this changing the hive size led to a huge difference in survival after 2 years. 83% for the small hive, 17% for the large hive. You might wonder what was going on with those 2 large hives that survived for 2 years. Those were some of the nastiest bees I've ever had. And they were so bad, I didn't even want to go into their hives at the end of this experiment. And it was painful to do that. I mean, it was painful to go into their hives. So I just parked them near the laboratory, several hundred yards from the laboratory, and just let them live. And they kept going on for years. Finally, they both died out, but they were very defensive colonies. And I suppose that defensiveness extended to mites as well, not just to humans. Next slide, please. The third guideline is to space your colonies as widely as possible. And I know this varies among beekeepers. Some beekeepers can space out colonies, some cannot. But here's, again, let me illustrate with it from an experiment, how this spacing, just spacing the colonies differently, changes things for the bees. This experiment involved, again, 2 groups of colonies. In one group there were 12 colonies that were crowded in an apiary. Field near the apiary. You can see in this image on the right, you can see where the apiary is labeled, and then you can see nearby there's this long field. It's actually an old radio astronomy antenna array, which I was able to get to use. And so in this long field, I was able to put the other 12 colonies with a more dispersed. So there was 30 to 80 meters apart between the colonies in the dispersed group were 30 to 80 meters apart. And I set these colonies up and I just let them go. There was no treatments for mites, but I did monitor the mite levels in the colonies. Next slide, please. We can go to the next slide. I want to emphasize there are 24 colonies in this experiment, and none of these colonies was treated for Varroa. Well, what happened? What did I find? Next slide. After 2 years, 0 of the 12 colonies in the apiary were still alive, and 5, but in the colonies in the dispersed group, 5 of those 12 were still alive. And what was going on here? Why this difference? What I found was there was very strong robbing of dying colonies in the crowded apiary. So some of the colonies started collapsing in the crowded apiary. Some of the colonies started collapsing during the first summer. And when those colonies started collapsing, their bees from the neighboring colonies would rob them out, and then the mite counts would go up in the colonies that were doing the robbing. And so it didn't take long for all of the crowded colonies to get high mite counts and die. But that robbing did not occur as strongly in the dispersed situation. So there was less robbing and less dispersal of mites among the dispersed colonies. So that's some of the evidence that we have that why dispersing the colonies, spacing them, helps them survive. Next slide, please. Third guideline, line your hives with propolis collection screens or build them with rough lumber. Again, here's the goal, is to get this, help your colonies, stimulate them to produce this propolis envelope, which functions as both waterproofing and an antimicrobial shroud, as Marla Spivak's lovely work has shown. Next slide, please. Fifth guideline, provide your most resilient colonies, the ones that are showing that they can control the mites. Provide them with drone comb. Give them 10 to 20% of their comb to be drone comb, because this is a way you can have your really good colonies. You can help them promote their genetic success. Every drone produced by one of these really good colonies is a way to help improve the genetics of the population of colonies in your area. Next slide, please. Sixth guideline is keep the nest structure intact. Whenever we take out a frame, put it back in the same location with the same orientation. There's a lot of beekeeping techniques that involve manipulating, spreading out the combs, and things like that to reduce swarming. But the bees have worked really hard to build the nest structure, so it's just the way they want it. And if you adopt this Darwinian beekeeping approach, one of the things you do is you want to keep that nest structure intact. So put each frame back in its original location with its original orientation. Next slide, please. Here, what I'm finding, and I'll explain this in some detail, it may seem like a small thing, but it turns out to be really important. Provide only a small entrance at the bottom of the hive. And it took me a while to learn this, but I should have learned it because I've been seeing for decades in the fall, the bees were plugging the top openings in their hives with propolis. You've probably all seen this as well. If there's a crack between the inner cover and the outer cover, they'll fill that in with propolis. And I'll show you why this is so important. What I use now is illustrated here. I give my hives a 2-inch wide entrance opening, and I put a 1/2 inch mesh, that's about 13 millimeter mesh, screen over the opening to keep mice out. So that's the entrance arrangement I have. Now let's look a little more carefully at why I give just a small entrance near the bottom of the hive. Next slide, please. This is an illustration that comes from the work of Derek Mitchell in England, where he's done these nice experiments where he takes an insulated hive and he puts inside the hive a 15-watt light bulb as a heat source. There are no bees in this hive. It's just an empty hive. And what he does is he compares the pattern of the thermal environments inside this hive with or without a top entrance. And what he finds is that if there's no top entrance, then there's a nice large warm area inside the hive at the top. Again, this is a well-insulated hive. However, if he puts a top entrance, as a lot of beekeepers, at least in North America, are taught to do for winter, that warm heat pocket in the top of the hive is much smaller and it's not as warm. So, and a lot of beekeepers, at least in North America, think, oh, I need to give the bees a top entrance, otherwise there will be condensation, lots of condensation dripping on the bees. And the other thing that has been found, it's not illustrated here, but you can see this, it's been found by a number of beekeepers, is that when they don't put the top entrance and the top of the hive is very warm, the condensation doesn't occur in the top of the hive. It occurs below the cluster, on the walls of the hive and on the combs below the bees. So the bees don't get wet. And what is also been found by other studies, including some of my own, is just how thirsty honeybees get in the winter. And so that condensation in the hive is the bees' water source in winter. Probably some of you have seen bees going out and collecting water in a conventional hive, even down to temperatures as low as 4 degrees centigrade. They're really pushing their limit because they're extremely thirsty in a well-ventilated hive without condensation. So ironically, that condensation is a good thing for the bees. Next slide, please. And so one of the things I have failed to do myself for many years is to give bees properly insulated homes. And you can see that in that example of the hive I'm showing there on the left is one of my Darwinian beekeeping hives. It's not well insulated, and one of the things I'm doing now is I'm building my hives with better insulation. I'm even doing an experiment this summer to put out bait hives, some of which are insulated and some of which are not insulated, and to see if the scout bees preferentially choose well-insulated homes when they have a choice. Next slide, please. And I guess this is my last guideline: refrain from treating colonies for varroa. And this is one of the hardest ones to do, but let's go through it. It involves monitoring the mite levels in your colonies, and say monitoring them at whatever interval you want, but often enough so you can tell when the mite level gets high. And if it gets high, more than 10 mites per 100 bees, or maybe you might even say more than 5 mites per 100 bees, then I'd say you have two options. You can either euthanize the colony, or you can treat it with miticides and requeen the colony with a queen of resistant stock. And the reason you want to euthanize, if you want to take that route, is it eliminates your non-resistant colonies. And by euthanizing the colony and taking it away, you avoid producing mite bombs, mite bomb colonies. We call them mite bombs here in North America, but I wish we would use the term robber lure, because it's not like these collapsing colonies are exploding mites. It's what's happening is robber bees from other colonies are going into the collapsing colonies, stealing their honey and coming home with mites. We've done experiments with genetically labeled bees to document that this is actually how the mites get spread when colonies collapse. It's not bees flying out of the collapsing colonies and moving into neighboring colonies. Almost all of the mite dispersal comes by robber bees going into the collapsing colonies and bringing the mites home. Okay, next slide, please. Okay, this is the next to last slide, and I include it because you might be wondering, well, what kind of colony survival, Tom, are you getting with your Darwinian beekeeping? Well, here you can see it. These are examples of some of my Darwinian beekeeping colonies, which I've got dispersed around. They're not in an apiary. One's out on the edge of the field, one's next to an old barn, another's next to a forest. I've been doing this for keeping track of this now for 5 years, and what I'm getting is the summer survival of the Darwinian beekeeping colonies is 97%. My winter survival is 84%, and so overall it's 81%. So that's a high level, you could say that's a high level of colony mortality of 19% over a year. It's much less than a lot of other beekeepers get, and you have to remember this is with no Varroa, giving the colonies no Varroa control, not treating them for Varroa, just letting the bees live on their own. And yes, so I lose about 20% of my colonies in the winter, but I then just replace the colonies with, where I am, I can put up bait hives and I can capture swarms, and I just rebuild my numbers in the spring. And what I'm finding is that my percent winter survival is going up and up, and this is gradually because I'm replacing non-resistant stock with resistant stock. So that's what I'm experiencing now. Next slide, please. I think it's the last slide. Oh yeah, I replace the losses by catching swarms. Oh, and the other thing I do is if I've got a colony that's really good, as it keeps its mites down very low and has survived the winter, I'll make a split from that and let the split rear its own queen. It's kind of an artificial swarming process, so that helps too. And last slide, please. So I just want to conclude by coming back to Charles Darwin, and imagine a little bee thanking Mr. Darwin for helping us understand the logic of this kind of beekeeping, and Darwin saying thank you. But I guess I do have one more slide. If we could go to that. It is to mention that there is this book that I've written, The Lives of Bees, and it's a good source of more information about this original environment, or the natural home of honeybee colonies. It's about how colonies live in the wild. And for those of you that are interested in thinking about this Darwinian beekeeping, that's the starting point, is knowing how honeybee colonies live in the wild, and using that as a guide for your own beekeeping. So, okay, that's the end of my talk. I'd be happy to address questions if we have time, and however you'd like to do, however you'd like to proceed.
Thank you very much. Great talk. I just love it. Thank you. And yeah, for sure, there are questions. There are actually quite a lot of questions. So I think we'll get in a very nice discussion here afterwards. I'm just sitting right now to copy the questions, so just give me a sec, and then I will be online with the questions. Like this. Okay, maybe I should just start from the top and give you some of the questions. And maybe we should start with a very hard question. I mean, you're talking about this, what to say, local bees, natural beekeeping, and then we have honeybees are actually not native to the US. What is your opinion about this, and when is actually bees locally adapted?
Yeah, that's a really important question. I'm glad the person asked that. Yeah, the Apis mellifera is not native to North America. It was introduced here about 400 years ago, 4 centuries ago. And so the way I'm looking at it is that gives these bees have had a lot of time to living on their own, to undergo natural selection to survive in North America. And there's different bees in different parts of North America. So that the bees that are living in the desert Southwest live differently than the bees up in the Northeast or in the Pacific Northwest. So it is a matter of locally adapted bees. But one of the things we know about natural selection is it can work very, very quickly. A good example comes right from the island of Puerto Rico, where Africanized bees got into Puerto Rico, and the bees were very nasty for the first 10 years, but because there are no natural predators of honeybees, in Puerto Rico, there was strong selection, natural selection against defensiveness. And now the Puerto Rican beekeepers find that those bees are quite gentle. So that's just one example of how quickly natural selection can operate. So I think our bees, wherever we are, in the bees that are living in the wild, they're experiencing natural selection, and they quickly adapt to their location.
Okay, because this was another question actually. How fast do you think that, I would now say, introduce bees that do adapt to the local conditions?
Yeah, I can't give a precise number for that, but I can say that with the levels of selection we're seeing, with those, for example, with those colonies headed by California queens, almost none of them survived. There were, so the selection is very, very stringent in the wild. So it's probably a matter of a few decades at the most.
Just to give you some information from the crowd, we asked out there, do any of you know about feral colonies that are kind of surviving for several years? And Pierre Krüger, who's actually the scientist at the Beekeeping Institute, he says that we do have feral bees in Denmark, and right now they're kind of trying to find out how doable those are. And they claim, there is a claim that in a lime tree, the bees are said to have been living since 23 years. Maybe that's not the same colony, but still. And the Danish Beekeeper Association some years ago actually made a questionnaire to beekeepers asking about, do you have any wild colonies out there living in a chimney or living in the wall of a house for a longer time? And they got some positive responses. And there was a lady who did react on this survey there that's talking about on one of our castles where they have a big wall up there and it's very, very high. Yes. And they have for 13 years been able to see bees flying in and out up in the top. And as he says, for good reasons, they have never been treated. So we have it on I think we have it on a low level, kind of. But how do you know that this is not kind of a very good nesting place for a swarm, and what we see alive every year is just a new swarm? And the same in your forest.
Yeah, no, those are really good questions. The reason I know that these colonies are surviving the winter, and it's not just a case of the nest site of a colony dying out and the swarm moving back in, is that I check these colonies 3 times a year, at the end of winter before swarming starts, in the middle of the summer, and then at the end of the summer. And each time I check a nest site, I look for bees bringing in pollen. That tells me that there's a live colony in there. If I don't see bees bringing in pollen, it could just be bees robbing out the honey from a dead colony. So it's that kind of program of, and I didn't talk about that at all in this talk, but that is what I find from those lines of studies, and I've done this twice now, for continuously for 5 years, is I find that colonies in the wild on average will survive for 5 years, and then the colony dies out for whatever reason. But meanwhile, during that 5-year period, the colony has been producing swarm. And I could go through that in detail, but that creates actually a balance, because if a colony produces a swarm every year, the swarm's colonies only have a probability of about 0.2 of surviving their first winter. So it takes about 5 years for a colony to successfully establish another colony that'll live on. But that's how it works.
Okay.
So that would be something really good to do in Denmark, would be for somebody to start monitoring these sites.
Yeah.
And you really have to think of it in terms of how long a site is occupied, because the colony inside there changes genetically every time the colony swarms or that replaces the queen. So technically we have to say a site has been continuously occupied. We can't say a colony per se has continuously inhabited. I'm sure you know what I mean.
Yeah, yeah. Wow, lots of questions are turning up. I'm not sure that we can make all of them. But here's one of the critical ones. And it was really, I think it's a fun question. Why did you actually in 78 monitor those wild colonies? What kind of interest did you have at that time? Who wanted to know this? Yeah.
Thanks for asking that question. I did that just out of pure curiosity. I realized that we had even though honeybees, nobody had ever measured the abundance of wild colonies, and that's all I wanted to find out. I just wanted to know, gosh, I know where this beekeeper has his hives and this beekeeper has her hives, but how many hives are there just that nobody's keeping? And so that's why I went to that forest. I went to that forest because I knew it was a protected area. It wasn't going to be logged or something like that. It was a long-term study site. I'm glad I did it. It was a lot of work, but it was good.
And one is asking also, not a critical question, but a curious question. How do you know that you did not miss out any honey trees out there? You simply surveyed that so many times that you are sure.
No, thanks again. I'm never claiming that I found all of the bee tree colonies in the Arnott Forest. All I can say is that, yes, so my estimates are minimum estimates of the density or abundance of the colonies. And in fact, I don't even survey the whole forest. I tried to make that clear that I really have it's a slow process, and I only have time to survey half the forest. And even in that half the forest that I survey, I don't claim to find all the colonies. So those are minimum estimates of abundance.
Great. When I'm looking on your pictures, I mean, I have to come and visit you there in Arnott Forest once. But the forest actually looks pretty uniform. I mean, the trees same kind of trees. Trees kind of have the same, I say now, thickness, the same heights. But it's an untouched forest for many, many years. So how is it?
It's untouched since the abandonment of the forest that started around 1880. Okay. And the last farmers left what is now the Arnott Forest in the 1920s. So it's been abandoned or unmanaged for about 100 years or more, depending on the part of the forest. The university, unfortunately, is starting to do some logging there, and that's dismaying to me, but I can't do anything about that. Fortunately, it's surrounded by state land, which is thoroughly protected.
I mean, my suggestion would be go and buy a very big iron chain and then chain you to some of the trees there that's going to be locked as a protest, and then you will see. Then you stop them.
One thing I wanted to say is, I don't know Denmark very well, but the time I spent traveling around there, I thought it's a great place for this bee hunting because it's not dense forest, and the bees are living someplace in the wild, as you pointed out. It would be really interesting just to go to one of the more remote parts and just see what you find in Denmark, because it looks like it would be a beekeeper's or a bee hunter's paradise.
Okay.
Wide open.
Maybe we should make a project and kind of encourage the beekeepers to do that. That would be really nice. There's one information here from one of the beekeepers that telling you that in Denmark the queen breeding is very well organized, and the main part of the Danish beekeepers, they buy queens from queen breeders in Denmark. And that's actually also my next question to you is actually, when do we talk about a local adapted bee? And you know, Denmark is not a big country, so could we say that all bees in Denmark or all queens in Denmark, they are locally adapted, since the country is, I mean, we have 40,000 square kilometers, so it's not a big country.
Well, if most of the queens have their roots in Denmark, then I would say you probably do have locally adapted bees that know about your seasons. Do you have a sense of what?
I have to say mainly is the Buckfast bees, which means that they use material that is introduced from other countries and other places mainly in Europe. So they make this hybridization of the Buckfast bees.
Well, that will be okay as long as it's they don't bring in. I mean, if it's ongoing, where they get some Buckfast and and the queen producers just keep producing from their best survivor stock, then I think that's going to give you a locally adapted bee.
Okay. Okay.
What I know in England, on the other hand, they import a lot of bees from Italy, and so they're really in a different situation.
So in one of your rules, you said that the natural bee colonies or houses, they're very thin walled, and it's kind of confusing a beekeeper, because we try to insulate our colonies the best we can do.
Yeah.
So
I'm sorry, I guess I didn't state that clearly. In the wild, their nests are very thick-walled.
Thick-walled? Okay, we misunderstood. I'm sorry.
Yes. In a tree, it would be 20 or 30 centimeters, sometimes even more, of wood that is around the tree cavity. And that's, fortunately, that's something that beekeepers can do rather easily, is provide their colonies with better insulation than
okay, so that's what we're doing. Yeah. There
was a question. How are beekeepers producing hives that are well insulated in Denmark?
Okay, but the main part of the Danish beekeepers are using styrofoam boxes. And then we actually have what we call, I don't know, in Danish we call it trosteder, which is a special Danish type of hive where there is, I would say, 10 centimeters of insulation in them. So that's the way to do it.
Yeah, that sounds good.
There's also a question, your natural bee colonies there, how often do they swarm? Do you have any counts on that? Do they all swarm once a year, or do you see swarming very seldom? Or?
Yes, the typical pattern is the colony will produce one prime swarm. The probability is about, I have to look up the numbers. The probability of swarming is, I think, about 0.9 in the colonies living in the wild. And they produce a prime swarm, and typically they will produce, I have to again look up the numbers, but a probability of, I think, about 0.5 of producing an after swarm.
I just give a comment to the beekeepers. Please do not post more questions. We don't have a chance to get through all the questions. I'm sorry, but it's there's somebody's asking, do you feed them, or do they just keep all the honey, or they keep the honey that they need, or the food? The honey, the feed that they need.
No, the colonies that I'm keeping with Darwinian beekeeping, I don't feed them. I just let them keep their own honey, unless I put on a shallow super, which I take off early in the summer.
And now you get the question back, how do you do to insulate your hives?
I take a Langstroth hive and I put, it is 1.5 inches, so that's about 40, it gives me it's polyisocyanate. It's foam that's 1.5 inches thick, and then I put that around the hive, and then I coat that. I put a layer of wood over the outside of that.
Okay. And then there was a question as well. I mean, when you keep your bees in this natural way, how often do you check your colonies then?
I check them 3 times a year.
Okay.
I check them in the spring. I check them in midsummer, and then I check them at the end of the summer.
And then there's one asking, actually, should we let the bees build their combs by themselves instead of putting in foundation in the colonies? Would that kind of improve something? That's the point.
Yes, when I do that. What I do though is I have a lot of old combs where the bees built their comb on foundation, so I use that. But and then what I will do is when I set up a new colony, I'll put in, let's say it's a 10-frame hive, I'll put in 5 frames of drawn comb, and then I'll put frames between those drawn combs with wires to support the new comb, and I'll let the bees build whatever comb they want in those empty frames between the drawn combs. Because in fact I do want the bees to build the combs just the way they want, and they of course, produce a lot of drone comb, which is, again, what I want too. Colonies that are, I want the survivor colonies to pass on their genes as effectively as they can.
Mm. So there's a nice comment from Sissel, he's from Norway, and we claim that he's the northernmost beekeeper in the world, actually. So we're really far away north of the polar circle. And up there, they really have to, I mean, they kind of double cover the colonies. And she's really telling that with this condensation, that she has the same experience, that she can actually see mold on the bottom board and on the side, away from where the bees are.
Yeah, it's seen bees desperately collecting water right at the edge of their thermal tolerance for flight taught me that the bees can get really thirsty in a conventional arrangement.
Yeah, and now here's actually also again with the bottom board. I mean, you have closed bottom board screens, don't you have that?
Yeah.
I mean, in Denmark we have inserts because of varroa. We want to count the varroa mites, and we actually keep it open during the winter to kind of keeping the moisture out of the colonies and keep them colder so that they kind of stick into the winter cluster. But you have closed bottom boards, isn't it, like that permanently?
Yeah, I do, and I haven't, and I understand the screen is useful for the varroa sampling. It seems like it doesn't seem to disturb the bees based on what the beekeepers tell me, and it's probably because this warm pocket is in the top of the hive. It's not the bottom of the hive. So as long as it's not windy, I think that having an open bottom doesn't, even though it's unnatural, doesn't seem to be too harmful for the colonies.
And then we have Chris here, and he really tries to understand why your bees are tolerant, and he's really asking, do you really know that the gene composition of the bees that you have there? And he's really asking how much percent of Mellifera scutellata do you have in your bees in Arnott Forest? And probably don't have any.
Yeah, well, we've done very careful genetic analyses of these bees, both the bees that were collected in the 1970s and the ones that were collected in 2010, and that gives us a very precise picture of their racial or composition. And back in the 1970s, they were, I'm going on memory here now, but they were primarily a mixture of Ligustica and Carnica. I think that's about 40%. And then the other 60% was 30% Apis mellifera mellifera, and the other 30% was Apis mellifera caucasica. And then when we resampled in 2010, done the same, and this is all from the Arnott Forest and the surrounding forests, we got the same percentages, except there were a couple colonies that did have a little bit of scutellata, about 5% in them. So it was, of the 30 colonies we sampled, it was 2 or 3 that had some scutellata. And that's probably because there are beekeepers that bring bees in from Florida, for example, up north for the summer, and they're bringing up some scutellata genes.
Okay, we're getting very close to 6 o'clock. So let me just handpick some of the most exciting questions. So did you survey your colonies on the hygienic behavior in any way, like Marla Spivak does? Or?
Yes, what we use the technique of looking for what percent cells have been uncapped and recapped, and this is building on this is the technique we've learned, or the assay we've learned from Stephen Martin. And what we're finding is we get about 50% of the brood cells, the capped brood cells, show us that they've been uncapped and recapped. And Stephen tells me that is diagnostic of colonies that are doing very well at using this technique for controlling varroa. But we also find when we put the colonies in hives with screened bottom boards, and we put sticky boards in the bottom, we find a lot of the adult mites that are missing their legs or have dents in their in their bodies. So it's not just the uncapping, recapping, which prevents the reproduction. It's killing the adults when they're out too.
Could you give comments on those special types of hives that we have? I mean, the Kenyan top bar hive, we call it also the African hive, or Varroa hives, or does that kind of give any sense or meaning in your way of keeping bees?
Yeah.
Do they help? Do they have any advantages?
I haven't used any of those hives, but from what I know of the Varroa hive, for example, that looks like a pretty good hive to me. It's a thicker walled hive. It has a small cross section and it's tall. So I think that's a pretty good hive. And I know of one beekeeper who does Darwinian beekeeping with Varroa hives, and all he does is at the end of the summer he takes a honey super off the top and he puts a new empty one at the bottom. That's his approach to Darwinian beekeeping. So I know those Varroa hives can work. The Kenya top bar hive, I just don't have experience. It would have to be a well-insulated hive.
Yeah. So I think I mean, we could keep, this is so exciting, and we could keep talking all the night, but we need to have people out to eat, get some food, because our next activity starts at 7 o'clock.
Okay,
so
very good. Thank you.
Sorry to the ones that didn't get their questions answered. I will publish them later on. And Tom, it was a pleasure. Thanks a lot. Hope to meet you soon somewhere out in the world, that we can start traveling again. Yeah. And
well, I want to thank the Danish beekeepers, all of you, for inviting me to share this information. And I hope that even if it's not directly useful, it might. I think it's always good to know how our bees, where our bees, what their natural lives are like. Good. So thanks again.
Thank you very much.
Wish you all well.
Same to you. See you next time. Bye bye.
Till next time. Bye bye.